Hi everybody, it's Mrs. Mancuso, and today we are doing AP Bio review, and so these are shorter questions, so these are more likely to be your question number 2. Usually they have 6 task verbs and are worth 6 points. The ones today that I found on AP Classroom don't necessarily have the same number of points because when they first made these questions, they weren't realizing that you were going to take it from home. But let's get started here. All right, students were using the formulas for the surface area and volume of a rectangular solid to model cells of different size and shapes. They produced the data shown in the table. So remember you guys did this, like you took the agar blocks and they were blue because they had bromothymol blue in them, and then you placed them in a container of vinegar and you saw how long it took for diffusion to occur in them, and you measured all of their lengths, heights, widths, and you calculated surface area to volume, and you definitely saw that the cells that had the fastest rates of diffusion had the highest surface area to volume ratio. And remember, you could even like cut them out in your own sort of shape and test it. So let's go through and do these questions. So number 1, identify the rectangular solid that represents the cell with the smallest amount of plasma membrane per volume. So smallest amount of plasma membrane means we're going to have a really low surface area to volume ratio because it means there's not much plasma membrane. So that one's 5, 6, 9. Okay, so it's definitely solid number 1. B, describe the relationship between the dimensions of the rectangular solids and their surface area to volume ratios. Okay, so every time they say relationship, I like to say as. All right, so let's look at, so the length is greater, the height is smaller, and the width is greater. So as the solids become longer, thinner, and wider, the surface area to volume ratio increases. So I want to take into account all 3 of these things, right? So when it becomes longer, thinner, not as high, and wider, and then the surface area to volume increases. Or is using the data from the model, evaluate the hypothesis that cells shaped like rectangular solids 1 and 2 are better suited for slow metabolism, and that long-term storage of energy containing molecules than 4 and 5. Okay, so if I have a 1 or a 2 versus a 4 or a 5, this would be better for storage because it doesn't allow diffusion as quickly. So solids 1 and 2 would be better for storage, less diffusion of molecules, or slower diffusion of molecules. 4 and 5 would be more efficient at moving molecules in and out. And if you want to store it, you don't want to move it in and out. So they are much better at moving it in and out. This is really slow moving in. And then the benefit is it would be very slow to move out, which is good because if you want to store it, you want it to stay in. All right, based on the trends shown in the data, explain why some rectangular cells include folds or projections. Okay, folds or projections increase the surface area to volume ratio of a cell, maximizing efficient diffusion without changing the overall shape or size of the cell. All right, I like that question. Next one, effect of environmental factors on DNA. Okay, treatment. Mean percentage of DNA with breaks in it. Okay, environmental mutagens can affect DNA in cells. BAP is a mutagen that is commonly found in urban air pollution. Researchers claim that the effect of UVA radiation, another known mutagen, is amplified by the presence of BAP. To test their claim, the researchers exposed cultured eukaryotic cells either to BAP, UVA, or both. The researchers then determined the percent of chromosomal DNA that had damage in the form of double-stranded breaks. The results are shown in the table above. Okay, identify the control. Okay, the control does not get my independent variable. There it is, untreated. So this is the untreated group. No mutagen added. All right, justify the need for a control. Okay, I need a control to have a baseline. Need a baseline to compare the percentage of breaks when there's no mutagen added. So we always have a control so that we can see like, all right, how much damage does it do when you don't have anything? Because like sometimes damages just happen. They're just mutations. So how often does it happen without the mutagens? All right, identify the dependent variable. So the dependent variable is what you measure in an experiment. So they were measuring mean percent of DNA with double-stranded breaks. The mean percent of DNA with double-stranded breaks. I always write it all out, exactly how they gave it in the table. And then the next one says independent variable. So independent variable is that one thing that you change. So you change the mutagen added. So whether it gets BAP or UVA or both. All right, using the results from all treatments, describe the effect of BAP alone and UVA alone compared with the effect of the combined treatment. Okay, let's see. Untreated gave me 9.8 plus or minus 2.4. BAP, wow, so it's a little bit less, but with this standard error, it could be a little higher, but it's right around the same. And 10.1, if I subtract 3.4, okay, so these are all the same. So no significant difference in the mean percent of DNA with double-stranded breaks if treated with BAP or UVA compared to no treatment. All right, now when you use both 35.1 plus or minus 5. Okay, so a large or a significant increase in the mean percent of double-stranded DNA breaks when treated with both BAP and UVA. All right, and you could even go ahead and put the numbers right in there. Like this one is this range versus this one is this range. That would only add to it. Again, write out what you can first, and then as you have time left to submit, think about adding things in there, especially if it says using the data. Go ahead and fill in the data differences um, that exist. So um, this is a super interesting study because those BAPs are mutagens that are in the air. So a mutagen can alter your DNA. So if it causes a break in the DNA, you're not going to be able to do transcription normally. So you're not going to be able to code for the proteins that that gene that was broken code for. And if you have them in combination, mutagens in combination seem to be giving a really devastating effect. So this is how some of those laws come about to control BAPs, because those are produced from burning different things. So like burning wood, even cigarettes, things like that release BAPs into the air. So certain chemicals, benzene chemicals. All right, I will see you guys today on the Zoom. Bye.